All right, thank you, and thank you all for coming. I told Bill I wasn't going to take more than 25 minutes to do the introduction, so it should be fine. I wanted to give you a little bit of background on Bill because he's such an extraordinary person. And I heard this story that he went to college at Yale. He was sort of pre-med, but then his aunt gave him a book called Atoms in the Family, My Life with Enrico Fermi. And that changed his perspective on things, and he decided to major in biophysics. So while he was at Yale, he took physics and biophysics course with Dick Setlow, who you may know was a later member of the biology department here at Brookhaven. After he's earning his BA in biophysics in Yale in 1958, he continued his studies at Caltech, where he earned his PhD in 1963, and then did a postdoctoral fellowship in biochemistry department at Stanford Medical. So during that time, when he came after that, he came to Brookhaven, and he's been here ever since. And during the years of 1990 to 1999, he was the department chair for biology, and he's also an adjunct professor in biochemistry at Stony Brook. So one of the things, his significant contributions contributed from the time he was hired until the present, he has more than 30,000 citations to his papers. He's among only 4 BNL scientists, including in the prestigious ISI highly cited category. So I wanted to talk about just 2 quick things from Bill. One, he's going to talk about the protein expression, so I'm going to let him talk mainly about that. But in addition to that, I just wanted to give you sort of a little background on that. In order to study the biochemistry and structural biology of protein, that used to be a very tedious and challenging protein purification process. And so for that reason, most of the proteins that were being studied were the ones that were in high abundance. Bill's basic understanding of the gene function of the T7 genome facilitated the development of the T7 protein expression system, which he's going to talk about today. Because of that, using that system, almost any gene could be expressed in E. coli to produce large amounts of protein for biochemical and structural analysis. This is really a game-changing development that's standard procedure in almost all biochemistry labs today. The other one that you may or may not know was that Bill developed a practical method for gel electrophoresis. He started that and had the instrumentation division here at BNL build something under his design in 1970 that was a great advance over the then current gel tubes, or tube gels. After a few weeks of using this system, he showed some results at Cold Spring Harbor, and everybody wanted one. So because of that, and because of Bill's nature, he was soon sending out plans on how to build them to hundreds of different laboratories. The companies rapidly picked it up on it, and now almost any laboratory uses his basic design, and the commercial has gone crazy. So anyway, in major awards, he won the Department of Energy's Ernest Lawrence Award, Memorial Award. He has also been elected to the National Academy of Arts and Sciences and the National Academy of Sciences in 1992. This is one of the highest honors that a scientist can receive. He's also a fellow of the American Academy of Microbiology and a fellow of the American Association for the Advancement of Science. One of the things that's sort of extraordinary is that Bill's patents have brought in more than $65 million to the laboratory over the years. And these ideas, and he's still working on new ideas, and he contributes to experiment new technology ideas. For example, in 2004, he earned the RD100 Award, and in 2009, the Science and Technology Award. He was also recognized by the town of Brookhaven at the first annual Inventors Reception. And in 2003, he was recognized at Stony Brook for the Faculty Honor Wall for recognition of outstanding academic accomplishments. And so in summary, I just wanted to bring this up. The reputation of BNL is really built on the accomplishments of true scientific giants like Bill. His ability to combine first-rate basic and applied research is truly remarkable. He also possesses a great deal of common sense, which we greatly appreciate, and an ability to cut to the basics of any problem. He's extraordinarily generous with his time and his advice, always being available to work with his colleagues in shaping ideas, developing proposals, etc. This generosity is especially helpful to young scientists, and I should mention that his editing prowess is legendary. It's a very great honor for me to introduce Bill for the presentation, Small Science, Big Impact, Basic Research with the Bacteriophage T7. Bill?
Thank you, David. Oh, that's great. Thank you. Thank you very much for those kind words. And I've got a lot to say, so I'm going to get right underway. One thing David didn't mention, at least I don't think he did, was that this month passed my 50th anniversary of coming to Brookhaven. So I arrived here on December 1st, 1964, coming from a postdoc at Stanford Biochemistry Department. And I came because, in my education, I always had aspired to be an academic and do research in a university and teach and so forth. But this opportunity to come to Brookhaven came along, and I thought, well, gee, I really like research. I will come here. They will set up my lab. They'll give me a technician. They'll give me support. I'll have a long-term perspective. And I will do the kind of research I'm interested in. And then I'll go to the university and be a professor. So that was my intent in coming. But you can see I'm still here. So let me just give an overview of what I'm planning to talk about. As David said, there were a couple of inventions with big impact. And earlier this afternoon, they had a gathering to appreciate inventors. And so I thought that would match pretty well. This topic would match with that. I also learned from my rehearsal with the lecture committee that physicists and accelerator scientists, if there are any of them here, don't necessarily grasp the vocabulary of biology. And so I thought I would give a quick overview of how proteins are made and what they're good for, because that's what these inventions are all related to proteins. And then I'll introduce Bacteriophage T7 and its host E. coli, just to give you a background there. And then I will talk about the basic research that we did. I couldn't possibly cover all of it, but the stuff that's related to these two inventions. One is slab gel electrophoresis. It's a tool for analyzing proteins, and the expression system is a tool for producing proteins. And this invention was the first one to go through commercialization at Brookhaven after the the Congress had passed the Bayh-Dole Act, which allowed contractors for government support to take title to intellectual property and to patent and receive royalties. I'll talk a bit about the commercialization and the problems in these also. And then I would like to just say a word or two about structural genomics and how it's related. So first, this is just a cartoon that I got out of a presentation that I made to the physics department 10 or 15 years ago, and I thought maybe it would be a good introduction. The genome of an organism is made of DNA. It's the double strand, the double helix. Has base pairs that go on for millions to billions of base pairs in a row, and it carries the information that's needed for any cell to grow. That information has to be read out in order to be used, and it's used mostly in the form of proteins, which are strings of amino acid that are maybe hundreds long to thousands long. And the first important enzyme for reading that out is called RNA polymerase. And I put that here because that name, RNA polymerase, and that enzyme is going to be important about much of what I talk about, so I just wanted to introduce it here. It is an enzyme that copies the base sequence in one of the strands of the DNA for each gene, which a gene gives the information for making a protein. So it reads it out, then the messenger RNA is picked up by ribosomes, which is a machine which just reads out that message and produces a string of amino acids that becomes the protein. And the information is coded in the base sequence in terms of 3 base pair triplets called codons. Each protein starts with a particular codon. ATG is the DNA designation, and ends at one of 3 codons. In this case, it's TAA here. And that string of amino acids makes up the protein. But that string of amino acids is really not functional by itself. It can only function after it folds up into a particular structure. And there are many, many kinds of structures, but the structure is needed for the protein to perform its task, which could be a structural task or a chemical catalysis or regulator or a signaling function. But any cell to grow has thousands of different kinds of proteins in general. So they are there performing all kinds of functions. And I'm just listing a bunch here so you get the idea that almost anything biological has an important role for a protein. So there's chemical catalysis for getting energy and regulating cell division or hormones. All these things in organisms of any degree of complexity. Cell walls, ribosomes are part RNA and part protein. Mechanical work, cell movement, cell division, muscles are proteins, and so forth. So I just make the point that they are the center of all biological processes. Now I want to introduce E. coli and bacteriophage T7. Now, E. coli is a bacterium that lives in our guts. It was originally discovered in 1885 by Escherichia, and that's why it's called Escherichia coli. He was a pediatrician who wanted to understand how does the intestine get colonized by bacteria in newborn babies. And he was the first to observe and isolate in the laboratory E. coli. It's a natural inhabitant of our guts. It's what's called a commensal. It lives with us all the time. Now there are the newsworthy variants of E. coli are the pathogenic ones that cause various kinds of bleeding or diarrhea or muscle wasting, all kinds of things. But those are the exception. Their main home is as a commensal in the gut, and they coexist with us. We help each other. That genome, the genome of E. coli, is about 4 million base pairs, and it specifies about 4,000 proteins. Now, T7 is much smaller. The bacterium is about a micron. This is about 60 nanometers and across. And it consists of a protein shell inside which is curled up along 40,000 base pairs worth of DNA. And that 40,000 base pairs specifies about 50 proteins. Now it's called bacteriophage. I don't remember whether it's, I think it's probably Greek, or anyway, that means eating, bacteria eater. And that was the name because when bacteriophage got into a culture that would be turbid of growing bacteria, and suddenly the whole thing clears because the phage has infected and broken open all these cells and the turbidity has gone, So it was initially named bacteria eater, bacteriophage. So in order to do all this, it has this little structure here, which we call a tail, which attaches to the E. coli cell, and then the DNA is delivered to the E. coli cell. And I was looking for some kind of diagram to show that, and it was only yesterday that I came across the drawings that I made for a lecture I gave, a B&L lecture I gave in April of 1970. And this, so those of you who are at that lecture, I'm sorry you have to suffer through this again. But this is the life cycle of T7. Here's the phage coming in, finding an attachment site. It delivers the DNA to the bacterium. And as soon as it gets in there, it has very strong signals that tells the E. coli RNA polymerase, that enzyme that makes messenger RNAs, to make messenger RNAs. And so it starts making messenger RNAs. Those messenger RNAs, now made from T7 DNA instead of from E. coli DNA, which is not shown there, they get picked up by the ribosomes and proteins start being made. Those proteins get to work and they start replicating the DNA. They start assembling themselves into structures that are called heads, which package the DNA. So the process continues, and now you start to get mature T7 particles. And after only 13 minutes at 37 degrees, the cell lyses, opens up, and releases a whole bunch of new bacteriophages, which can go off and infect more E. coli cells. So this is a very active and virulent phage because it in fact uses whatever's in that cell and converts it just to making new phage particles. Once a cell gets infected, it never escapes. So the question is, what are all these proteins doing? And the way to find out that kind of thing is to make mutations. And a mutation is a change in the nucleotide sequence that, that makes a change in the function. So the particular kinds of mutations that I started out to isolate were called amber mutations. They were quite new at the time. And what they are is a mutation in a codon. Here's in the normal protein, this codon makes glutamic acid. And the mutation changes this G to a T. And now it turns out that TAG is another termination signal that tells the ribosome that's the end of the protein. So the ribosome only goes and makes part of the protein. So in amber mutants, those proteins will be defective. And now you can study to see what were their roles in the infection. But of course, if they're an essential protein, you can't grow the mutant. Well, you can, because there are also what are called permissive hosts that I'm not going to go into the details, but they allow the ribosome to read through that termination codon and go to the normal end. And so you get enough of the protein to support the growth of the phage, so you can grow the mutants in the permissive host and study their lack of function, what the lack of function does in the normal host. So that is what we did. And so this is an agar plate showing what plaques look like. Anywhere an active phage is, it starts to grow. And the plate itself is spread with bacteria. And so as they grow and the phage grows, it eats them and forms these things called plaques. And each plaque is due to one initial phage, and so you can count the number of phages you have. And since we have two kinds of plates, one with permissive and one with non-permissive hosts, we can just simply by isolating mutants that grow on the permissive host but not grow on the non-permissive host, we can isolate amber mutants. And we did that. We isolated more than 400 through genetic manipulations, which I'm not going to go into. We could assign them to one of 19 different genes, and 104 of those mutations were made into a genetic map. And what I have showing here is Olive Ritter. The picture was taken in 1977. The pictures that I have are all from one of our albums that we had a bunch of pictures, and so that's the only ones I have. But at any rate, Olive was my technician for I'm not sure how many years she was, but for a long time. And she did an awful lot of plating of bacteriophages and counting plaques in order to be able to generate this genetic map of T7 amber mutants. I don't mean for you to go into any detail here. Each solid line is a gene, gene 1, and I numbered them purposely from left to right so you could tell the gene position from its number. And so there are 19 essential genes here, meaning that amber mutants wouldn't grow on the non-permissive host, but they would on the permissive. So this kind of map could be made. It took a lot of work, but we got a good collection of genes identified with amber mutants in each, amber mutations in each, and so we started looking at what were the functions. And one thing you can look at is, do they make DNA after they inject their DNA? And the first 6 would not make DNA or had some defect in making DNA. So those were proteins that somehow are involved in replication. But the real tool that was most effective here was being developed independently by Jake Maisel at Einstein College of Medicine. Jake and I overlapped in graduate school at Caltech, and so when I came to Brookhaven and he was already at Einstein, we got together, and I learned that he had developed a method that measures the length of a protein. So all my mutants had a shortened protein, so they would be missing from a pattern of proteins that were separated by electrophoresis on a polyacrylamide gel. And I won't go through this in great detail except to say that the detergent SDS negatively charged binds lots of copies to the proteins and unfolds them. And so now that complex of SDS and protein all has about the same kind of a shape. It all has a negative charge. If you put it in an electric field, they will migrate through this gel, which is just a matrix that's usually 75 to 95% water. So it's full of channels. When you force it through there, the channels are different sizes, the proteins have different sizes. The small proteins can go through any channel, so they go rapidly through there. The big ones can only go through the big channels, and so they go slower. So in the end, you separate them by size. And this is a picture of that Jake made. Now, I should say, I didn't complete my story there, that so Jake and I got together. We realized that this would be a good collaboration. And so I had the mutants, he had the method, and he came out with his family the summers of 1967, 1968, spent the summer at Brookhaven, and we collaborated on these experiments. So I had the mutants, and we could label them specifically with radioactive amino acids. The host cells would not be labeled because we gave them a short burst of ultraviolet light before infection, so they couldn't make their own proteins anymore. But when the undamaged T7 DNA came into the cell, they would make T7 proteins. This particular gel as all our early ones, is represented as migration upward. So these are the big ones, and those are the smaller ones. The whole rest of the world did it in the other orientation, so we soon switched to that orientation, and the rest of the ones I show you will be in the other orientation. But the thing I want to point out here is, so here's the cell with no T7DNA. This is the wild type. Everything is competent. You start to look through these things, and you can see, well, there's one that's missing here. So that's the gene 16 made that protein. There's another, I'm not seeing them so clearly. Here's one. You can look through here. And so here's one for there. You can look through and see, we could identify most of the proteins. This gel resolves big proteins, and another gel resolves smaller ones. And so we could actually resolve most of the gels. The most interesting one on here, I want to point out, you probably don't even notice it, is gene 1. It only makes one protein on this gel. This is the gene for T7 RNA polymerase. T7 RNA polymerase is needed to make all the other proteins. Only a few proteins can be made by the E. coli RNA polymerase. Most of them are made by T7. So this, by 1969, we published I published 3 papers, one by myself, one with Jake, and one with another person who was working with T7 mutants. And we could, at that time, we could say something about the order here. I numbered them in order. And that gene 1 we knew was regulating all the other genes. We didn't know at that moment that it was RNA polymerase. That was only a year or so later that Mike Chamberlain at Berkeley discovered it was an entirely new polymerase, different from anything anybody had known. It was very active, but it worked only on T7 DNA because the site at which it would initiate RNA synthesis was very long, 23 base pairs. So it wouldn't be found. It's highly conserved in T7. There are 17 sites in T7 DNA, but no sites that can be used by E. coli or by T7 RNA polymerase in E. coli genome. So it was very specific for T7. Let's see what I've got coming up. Okay, so the next thing was, at about this time, Einstein offered me a position, a 10-year position there. And I, in fact, accepted it. I had expected to be an academic and go be an academic. And so I was getting set to leave for Einstein. But as it happened, Einstein has periodic fiscal crises. They had no endowment. And so it took them a year and a half to get through the tenure appointment. In the interim, the biology department doubled my personnel from 2 to 4, myself, my technician, and I got, that's the one that was up here, Gary McGovern and Martha Simon. Gary's a technician, and Martha was a postdoc doing EM mapping of D7 deletions. So my personnel doubled, and so did the lab space. They gave me another lab space. They gave me tenure. And so by the time the Einstein tenure offer finally came, I decided I would stay put. And I never regretted it. So the next story, next part of this story is the development of slab gel electrophoresis. And the reason that came about was because I continued to collaborate with Jake with samples, and he would prepare them and run these. Now that was not a simple task. These each one was grown in a glass tube. Was run in a glass tube. It had to be extracted somehow. Then he sliced them longitudinally with something like a boiled egg slicer. and then lay out each sample on a filter paper, dry it down, and then put it next to X-ray film to get this image, an autoradiogram. So when Jake went off on sabbatical to Cambridge, England in 1969, I was stuck with doing this myself. And after some period of time, and Gary McGovern was also then working on it, I said, this is way too hard, and there must be something easier. And Bill Siegelman, at the time, pointed me to a paper by two Australians, Reed and Bolesky, who had described a vertical slab gel system that would have essentially the same orientation as these. I was looking for something that had a vertical electrophoresis pattern. The problem with their device was it was very difficult to load samples and kind of complicated. And so I started with their basic part and modified it to make a slab gel system. And now I just wanted you to compare these patterns, which have lots of surface noise from the radioactivity dragged across the surface of the gel that aren't always in register very well, with the slab gel, where things are pretty much in register. You don't have the surface noise. It's very easy, much easier to see which ones are missing in this pattern, and also peptides that were not full length and that kind of thing. And plus, it was so much easier to use. This is the picture of the slab gel apparatus, and that was made for us in the electronics shop at Brookhaven. According to, we kind of worked it out together and got the design. There are two glass plates here that are with spacers, and the liquid acrylamide is poured between the plates. And this plastic comb that has teeth sticking down, and the front glass plate has a notch in it that holds the comb. After the gel has set up, you pull that out, and then you have 25 slots to put in samples. You affix it to this thing, pour the buffers in, turn on the juice, and they migrate. And then when you're done, you just take them out, dry them onto the filter paper, and autoradiograph them. So that was a huge reduction in effort and a big increase in output. And it just happened, as David said, that I went to Cold Spring Harbor. It was just a week or two after we got our first results. And I told them, and of course, everybody loved it, everybody wanted it. People come through Cold Spring Harbor all year round to meetings, and the information spread rapidly. We made plans, which they handed out to the people who came there. I mailed out probably 1,000 of them, at least hundreds, and including to companies. And so it was, it was done in 1970. The first published picture was in 1972, published by me. And the apparatus itself was described in the paper in 1973. So by the time this paper came out, it was already in wide use, being offered by companies, and it had a huge impact in the field. And as David said, any lab who works with proteins has one of these things in it. And of course, each company, they didn't just copy this, they made, their own modification to make it look great or whatever. And another thing that is that the Human Genome Project, probably the first 2/3 of the Human Genome Project, the DNA sequences were analyzed on large slab gels of exactly the same design. So it was also a big part of the human genome. So that was a huge impact, and it was before the days of the Bayh-Dole Act, and at this time there was no incentive for anybody, any national lab or anybody, to patent anything, because nothing would ever come back to the lab or the inventor. It would all just go into the Treasury, not even to the DOE, or at the time it was the Atomic Energy Commission. And so it was easy to give things away and let them go. And in fact, I love to do that anyway. I think if something's useful, people should have it. So now I want to continue with a little bit further into the carrying us from 1969 to 1982. John, a big acceleration of activity came when John Dunn joined the department in 1972. Independent position there, but we collaborated right from the beginning. He came as from a postdoc and a graduate experience as a biochemist. He was expert in E. coli RNA polymerase, but now also in phage RNA polymerase. He had made a very significant discovery, which I won't go into, as a graduate student about E. coli RNA polymerase. And so we collaborated right from the beginning. And John was fearless in adopting new technologies. And when DNA sequencing became available, sequenced the entire T7 genome, which allowed us to map all the information we'd been getting. Another big advance at the moment, at that same period, was Alan Rosenberg in the lab, who got very interested in cloning and mapping T7 genes, physical mapping, cloning. He was really developed into lab manager, was a mentor for other people in the lab, led several different projects and collaborations. So we now had developed a somewhat larger group working on these things. And I want to just summarize the state of our knowledge at the point when the sequencing was completed. And here's the map, physical map, with the links of the genes. These are three regions. This region is the early genes, which take over the host cell. These are the DNA metabolizing genes, and these are the phage particle genes. The T7 RNA polymerase gene is here, and the T7 RNA polymerase just recognizes these things with a phi in front of them. That's where it starts RNAs. These are the RNAs that are produced down here, these by E. coli polymerase, and all of those by the T7 RNA polymerase. But this is my favorite picture of how T7 works. And it is a time course of protein synthesis during T7 infection. So these are pulse labels, 2-minute pulse labels to show what's being made at that time. Not what's there, but what's being made. And so this first one is just E. coli. It's making all its own proteins. That continues for about 4 minutes. These are 30 degrees to lengthen the time scale, so to spread things out. But then you see, here comes some T7 proteins being made. This is the T7 RNA polymerase. It's made only for a short time, but it's very active and stable. So the whole, at this temperature, the whole process takes only 25 minutes or so. So there's first the early genes, then come the DNA metabolizing genes. These are the ones that kind of shut off in the middle. And the DNA capsid genes, the late ones, go all the way to the end. The important thing here is that T7 is extremely efficient at shutting off the host gene expression, directing all gene expression in the cell to T7 DNA. Now, we weren't the only people who had seen this kind of thing, and there was, in fact, a lot of interest in cloning the gene for T7 RNA polymerase. That's for two reasons. One, the purified protein was being used to make RNAs in the test tube for all kinds of things that people were interested in. And so purifying the protein that's only made for a few minutes during infection was a big problem, and it's getting a lot of it. So people would like to have a clone of it, so it can be produced by itself, and you purify it more easily. But there was another, companies were interested in, and people who were making, was interested whether you could make an expression system driven by T7 RNA polymerase instead of E. coli polymerase, which all the ones in E. coli were all driven by E. coli RNA polymerase, whereas T7 RNA polymerase is much more specific just to the target protein, nothing else in the whole cell, and it's 5 times faster than E. coli RNA polymerase, and it doesn't pause. It goes through any sequence that you give it. So, in fact, so there was interest in cloning, and companies and other people thought that would be a good idea, and I thought it would be a good idea too. Other labs had tried and failed to clone the gene. It's a long gene. One lab reported cloning the gene in 1981, but it gave no active polymerase. Now, we knew so much from the basic research about the T7 that I looked at the thing and I thought, we had, thanks to Alan, considerable experience in cloning T7 genes that are toxic to E. coli, and I thought I knew why the gene wasn't being cloned. About that time in 1981, Perry Davenlew was a new postdoc, and when I told her about that scheme, she was very enthusiastic about cloning the gene, and with Alan's help, she succeeded in February 1982. Now, these diagrams, I want to just go over quickly, that the gene, here's the gene cloned in this direction. The sequence, John had sequenced this whole region by that time, and we could see that there were 2 what are called promoters, places where T7 RNA polymerase starts synthesis. Well, it's hard to, by the technologies of the time, you just had to cut with restriction enzymes or something else to get a chunk that you clone. Every one of them would have been very hard to separate this, which is only about 20 nucleotides from the end of the gene. And so every clone would have a promoter for the gene. So if you even got a molecule of it, it would start making its own gene, and it would just autocatalytically take over the whole cell. Its cell would not be able to grow, and no wonder nobody could get a clone. So our strategy used T7 deletions, things that had been mapped by Martha, and make heteroduplexes that have single-stranded loops that can be degraded by a specific enzyme to give a fragment. And so taking these and then digesting with another nuclease that digests from the ends. Perry did this. This was a hard job, and screening. Perry and Alan were screening by various methods, and finally came up with a clone. That was PAR. This, by the way, this is lab conventions around all the time, was Alan Rosenberg. So all our plasmids and vectors that we made are PAR, and they just keep numbering up. So at any rate, we could test easily. This gene was functional. We made a copy that could be expressed from an E. coli promoter under control of a repressor, so that we could make large amounts, and so that was all that was a big success. But when we tried to express target genes using that T7 RNA polymerase that was provided by that plasmid, all those efforts failed. And it's probably because that was a multi-copy plasmid and there was too much basal activity before induction to tolerate a T7 promoter in the same cell. So to reduce that expression, we took a single copy of that module from 1219 and inserted it into the bacterial genome. And this was done, the construction of the vector for insertion was done by Barb Moffett, who was a PhD student in the lab. She transferred that module into a lambda, phage lambda, a different kind of a phage cloning vector, and lambda integrates into the chromosome. So then we could integrate into the chromosome as a single copy. So now there would be only one copy of the gene, and the basal expression would be much lower. That host was called BL21DE3, and it's used around the world. So we got successful expression in May of 1983. So this got us the polymerase to do the expression, and so here is the result. Before expression, that's, okay, these are stained gels now. So they're saying what's there, not what's being made. So before induction, this is the target protein, and it's made in two forms. There's very little, if any, that you can see on a stained gel, but 30 minutes, 1 hour, 2 hours, 3 hours, it's made so efficiently that certainly more than half the total proteins in the cell are the target protein. And this part shows the RNA, that there's enough RNA to saturate all the ribosomes. So these are ribosomal RNAs, this is the target DNA, RNA. So it was extremely effective, and people were very interested in using it. Then we got into a flurry of vector making, which I won't go in detail, but we called them PET vectors for plasmid for expression by T7 RNA polymerase. And their different iterations were made up to PET 11 in our lab, and the people who made them were Alan and Daoshan, John Dubendorf, Barb Lade in John's lab, and Shubha Lin, who was a visiting scientist in John's lab at the time. And so we published papers on the vectors. This is simply a diagram of the system, of a system that's inducible. So the part up here is in the genome of the host cell. It's got an inducible promoter for T7 RNA polymerase. The T7 RNA polymerase that's made comes out to the T7 promoter, where it initiates transcription into RNA synthesis of the target protein. And there are other signals of T7 that have effects like stabilizing the messenger RNA or making it very efficient at being recognized by ribosomes and translated. So that was very successful. Now this was done in the period covered by the Bayh-Dole Act. So Bayh and Dole were senators, and the legislation was enacted in 1980, end of 1980, allowed universities to take title. The Department of Energy was not very interested in giving patents, and so in 1983, there was a presidential memorandum that directed all federal agencies to extend Bayh-Dole to all government contractors. So at that point, BNL had the mandate to patent and commercialize inventions. Part of the Bayh-Dole Act said that inventors must share in any revenue received from licensing, and that any remaining after expenses had to be used to support scientific research or education. And so in this period, Peg Bogosian was a patent attorney, came to head the tech transfer office, and she came around looking, asking people, do you have something that's looks like a good idea? And so we told her about this, and the first patent was which was for cloning the gene, was filed in 1984, and now came a long period of frustrating back and forth with the patent office. They were this was rejected went through many cycles of rejection. And during that period, a continuation was filed. But in fact, we finally, Peg and I, had to go down to Washington to meet in person with a patent examiner to explain why what we were doing was not obvious. And in fact, the first patent issued in 1990, that's 6 years. I just point out here that the delays were actually beneficial because that time patent expires 17 years after date of issue. And this is kind of the patent history. We first filed, it was abandoned. This is the first one we got. Then there were more that were rejected, abandoned, and finally until we got another one here. And this is the one that counted the 17 years from. And so it expired 2 weeks ago. So we had the long benefit of royalties from those patents. Now, there were problems in commercializing. And they were self-inflicted. Once the paper describing the system got published in 1986, and I started receiving requests for it, and it's freely available to academics, even though we were trying to patent it at the time. We wanted it to license for commercial use. We didn't want to impede any research. Several companies inquired about distributing these materials to scientists. However, this, as I said before, was the first technology to be commercialized under Bayh-Dole. And the obstacle came from the board of AUI, the contractor who administered Brookhaven Lab. They were mostly physicists. They didn't understand kind of the way things go on in the biological community. They said, we're not a for-profit. We don't want the profit, but we don't want risk. And so they wanted liability insurance of $5 million, and these small biochemical supply companies really couldn't obtain it. So meanwhile, I had to devote one technician full-time, and this went on indefinitely to send kits to researchers. I probably sent out 1,000 kits just from my lab. This load was finally lifted when Novigen was a small startup, but backed by a large company I had never heard of. And the company said, oh, we'll self-insure. And so Novigen got the jump on everybody else, and they had it to themselves for about a year or two, in which they made the most of it. They made many different pet vectors and modified hosts, and they really made a big success. It made the company. They wanted an exclusive license, but we would not give it because it's a research tool. And ultimately, the insurance requirement was reduced. Now, many companies got it. The system expresses, well, anyways, used around the world, is licensed by more than 1,000 companies. In order to get companies to use it for their own research, we had a very nominal fee, I think $1,000 a year or something like that, to use it for research purposes. And then, of course, when something commercially valuable was developed, then there were licenses on the marketing of it. So that was the biggest part of royalty. So that's that part of the game. It's hard for me to see the clock, but I think I have enough time to just say briefly about structural genomics. The sequencing of the human genome, what's that? Well, maybe I should just skip that part. I don't know. Here, I've got a thing here. We're going to wait. We're going to wait till log off. Okay. All right. So at the end of the Human Genome Project, the price of sequences is coming down. There were new technologies for DNA sequencing, more and more whole genomes were sequenced, many protein, millions of protein sequences that you could just translate from the DNA sequence and know what the amino acid sequence of the protein is, but we didn't know for most of them. And you could make families of proteins, but many families, thousands of families of proteins that are obviously related and would have the same structure and probably a similar function, we didn't know about. And so taking the Human Genome Project as a model, structural biologists proposed, let's have a structural genomics project in which we scale up all these technologies that have been developed to an industrial scale, and we turn out a lot of protein structures for each family of unknown structure or function, and we build up a database of structures and functions that ultimately we'll be able to model structures and functions right from the sequence itself. And the plan then was, this was the steps in high throughput structure determination. You get the informatics, find out which families are interesting, you clone and express, and this is where T7 came into this thing, is the cloning and expression. We did a pilot project when this was under consideration of 100 or so, more than 100 yeast proteins, and we expressed them, purified them, tried to crystallize, collect NSLS, collect X-ray data, and produce structures. That lobbying was, and workshops and all was successful in 2000 when the Protein Structure Initiative was funded by NIH. One thing that in the workshops encouraged people to think this would be successful is there are many different experts in all these different steps, and each one of them said, well, the bottleneck is not in my part, it's somewhere else. And so everybody who was knowledgeable about anything thought the technology had reached the point when this would be possible and desirable. And so, in fact, it got underway. And at Brookhaven, we were part of a big New York consortium. And here's Swami and Eshwar and Kumran. They determined more than 400 structures by X-ray crystallography at the NSLS. Now, the proteins that they used were mostly came in this consortium. They automated the T7 system in other sites. And so the purified proteins, they were cloned and expressed at other places in an automated fashion, and then the proteins would come here and they would determine structures. But also I was a part of that initially and started wanting to make new vectors for high throughput cloning, and we have developed them. Eileen and Sue Ellen. Sue Ellen was here for the initial discussion and construction, and then she went off to Chicago. Eileen has continued until now. We have now vectors that have a much wider range of capability for maintenance and expression than PET vectors, and we can express easily, co-express 3 or more proteins, which is something that structural biologists are getting more and more interested in, is protein complexes as opposed to individual proteins. The last thing that I wanted to show here was autoinduction. This is something that I developed because to produce lots of proteins in parallel, you would have, in the original way, you would have to grow each one to the proper density, culture to the proper density, then add the inducer, which would induce the synthesis. I developed a medium that would do it automatically. And I won't go into details, but it's also been patented and commercialized. Using autoinduction, you can just put as many in parallel as you want, inoculate them, grow them up to saturation, and they will all be induced. So that's, again, were things that developed because of the interest in this scale-up. So I've given you the kind of stories for 2 of these high-impact things. We started out, our interest was in basic research. We weren't looking to make commercial impact in our current climate. Lessons learned is a good thing here. That so the things I have here is I think that small-scale, curiosity-driven basic research can really be compelling for the person who's interested in doing it. It's not necessarily possible to predict what impact that research may have, but high impact results are possible, and we had 2 or 3 of them from our work. And the conclusion I would make is that long-term support of small-scale basic research in areas relevant to DOE emissions is well worth the investment. Thank you. Any questions?
We have just a little time for a couple questions, and then we'll have refreshments out in the lobby. So is we have 1 or 2 questions anybody'd like to ask?
Bill, over the years, are there any areas that you were too busy to address that you kind of wish you'd have done?
Well, yeah, there's always things that you might have thought, but I think that mostly I just followed what was my interest at the moment, and so there are things even now. Occasionally I think, gosh, I should go back and work that out. I don't suppose it will happen anymore, but anyway, we did a lot, and of course I only covered a small part. Synthesized a lot of things that went into those conclusions at the end. Alan?
I can only imagine you have an awful lot of thank you notes, people saying thank you for your kits, but I think it's important to realize and emphasize the open attitude you had towards research and sharing, and I think that's a remarkable thing, and I want to say thank you for that attitude.
Well, thank you, and I think that research tools shouldn't be exclusively licensed. They should be widely available. And this is a real case in the point because it got so widely available and people develop all kinds of things with it, and you never know what's going to be valuable, but you catch them all. So I should say that sending out those kits was an evolving process. First of all, they said, okay, it's a patent. If you're going to give them stuff, first send them a letter, they have to sign it, send it back, and then you can send it. Well, that went for the first 100 kits or so. And I said, look, can't we just send them a notice along with the stuff and say, there's a patent involved if you do develop anything commercial, that you could freely give it to anybody, any non-commercial lab. If you were going to give it to a commercial lab, then they have to have a license. And so if you give it to anybody, just give this notice along with it, that's okay, so that the people are aware. I mean, the patents are in force anyway, whether they were aware or not. And so, okay, we got to that point. So then we're just folding up that thing and send it out with every kit. But even so, there were something like, I don't know, 6 or 8 auger stabs that were all wrapped up in a padded envelope, and so.
If anybody has
Thank you.